Hey everyone, in this video I am going to talk about RNA polymerase II and how it transcribes not only messenger RNAs but also these non-coding RNAs called small nuclear RNAs, and these small nuclear RNAs are going to be important for the main topic of this discussion, which is exon splicing. So splicing together those exons and removing introns from nascent pre-messenger RNAs. So to rephrase that, so RNA polymerase II, we've already talked about how it transcribes messenger RNA encoding genes. It also transcribes small nuclear RNAs. Now, specifically, it transcribes 4 out of 5. Having trouble spelling out. Out of 5 of these small nuclear RNAs. So there are 5 types: U1, U2, U4, U5, and U6. So the U stands for uridine. So these are, or uracil. These are uracil-rich RNA molecules. And the last one here, the U6 small nuclear RNA. So this one is transcribed by RNA polymerase III, and we're not going to talk much about that one. So if we cover these 4 right here, you'll be able to see, learn a little bit about how these are involved in exon splicing. So in previous videos, I covered sort of the transcription cycle for RNA polymerase II. And if you recall, there were about 5 different stages. So recruitment of RNA polymerase II, initiation. So recruitment is recruitment to the promoter by recruitment of RNA polymerase II to the promoter by the general transcription factors. Initiation, which would be unwinding of the DNA template and RNA polymerase II starting to transcribe the template, and there's abortive initiation too. So it takes a while for RNA polymerase II to get stably engaged. So we also have early elongation. And okay, initiation was less than or equal to 12 nucleotides long. That's referring to the nascent RNA that's being transcribed. Early elongation, that is let's say 13 to 60 nucleotides long. So the RNA would be somewhere in the range of 13 to 60 nucleotides. And this is biology, so that's just the range. Of course, sometimes it can be longer than that, but for the most part, early elongation is characterized by an RNA polymerase that has a nascent RNA of 13 to 60 nucleotides long. Then productive elongation. And number 5 is going to be poly A addition and termination. So now those stages right there, those are the typical steps or stages RNA polymerase II goes through when it's transcribing a messenger RNA encoding gene. Now, when polymerase II, POL2, I should say, is transcribing an snRNA encoding gene, the stages are somewhat similar. So let's say recruitment is going to be somewhat similar, and initiation and early elongation. So now if you remember what happens during early elongation for the messenger RNA encoding genes, well, the 5' end of the nascent RNA is capped. Yes, so capping occurs also here during early elongation for the small nuclear RNAs. So, however, the fourth and fifth stages are going to be very different. So these small nuclear RNAs are rather short. So we're not really going to have a productive elongation stage. And there's going to be a big difference when it comes to cleavage. So there's going to be no poly A tail, and there will be termination. So we're going to go with 3' end processing and termination. So the transcription cycle is a little different for RNA polymerase II, depending on whether it's transcribing a messenger RNA encoding gene or a small nuclear RNA encoding gene. And I just want to touch on this right here, how that termination stage, that last stage, is going to be different. So if this is RNA polymerase II, here's the DNA template. Here's the nascent RNA, and let's assume that RNA polymerase II is transcribing an SN, a small nuclear RNA encoding gene. So for these types of genes, there's a special complex that rides along with RNA polymerase II, that is thought to ride along with RNA polymerase II by interacting with certain modifications on the C-terminal domain of RNA polymerase II. Now the C-terminal domain, which is something we covered in the previous video, is diagrammed in yellow. And I'm going to put special phosphates here. So this complex is going to be called the integrator complex. And what it does for small nuclear RNA encoding genes is it scans the RNA. I'm going to have to diagram the nascent RNA over here on the right here. It scans the RNA for a sequence called a 3' box. Now the 3' box is to the integrator complex. Let me spell this out for you. Is to the integrator complex as the polyadenylation signal is to the CPSF complex for messenger RNA encoding genes, right? The CPSF complex, remember that was the complex that watches out for the polyadenylation signal, and when it sees it, it cleaves the messenger RNA. So what the integrator complex does is it scans that nascent RNA for a 3' box, and when it finds it, it cleaves the nascent RNA about 20 nucleotides upstream of the 3' box. So for messenger RNA encoding genes, the CPS complex, what that did, that cleaved the nascent RNA downstream of the PAS. So the integrator complex, that cleaves the nascent RNA upstream of the nascent RNA. So you end up with something like this. RNA polymerase, DNA. I won't diagram the C-terminal domain or integrator. I'm just going to cut it right there. And that gives us a 3' end and a 5' end. Now, that free 5' end can be used by an exonuclease, such as XRN2, which can degrade that, and when it makes contact with the RNA polymerase, it can cause RNA polymerase to dissociate from the template, thereby terminating transcription. And we have, as a result, we have the pre-snRNA. And like I said, this has a cap, 5' cap. At first, the cap is just like a cap for messenger RNA, 7-methylguanosine. It was made with the capping enzyme. And during processing, this cap is going to be changed. It's going to be methylated some more. It's going to become like a trimethylated cap. But this pre-snRNA now, it's going to go through a number of processing steps in the nucleus. It's going to be exported to the cytoplasm, where it goes through a number of other steps. Then it's going to go back into the nucleus. And during this process, it's going to associate with a number of proteins. So eventually, it's going to be called an snRNP. So what that is, that RNP stands for ribonuclear protein. Nucleoprotein, I should say. Ribonucleoprotein. And essentially what an snRNP is, it's a complex containing a small nuclear RNA and a number of proteins. So, what those processes are going to give us, RNA polymerase II transcribing these different small nuclear RNA encoding genes, and then those small nuclear RNAs being processed into the snRNPs, what we're going to get is a U1 snRNP, U2 snRNP, U4, U5, and then we'll also have a U6, but the U6 goes through a little different processing step, and it's transcribed, the snRNA is transcribed by RNA polymerase III. So just to show you, or have you get an idea of what these things, what their characteristics are, you know what I mean? Are there hundreds of proteins or a few proteins? Well, taking a look at the U1 snRNP, so it contains the U1 snRNA, which is about 150 nucleotides long in humans. 7 proteins called ring proteins, and 3 other proteins that are specific to U1. So 3 U1 specific proteins. So that's one snRNP. Now, let's take another one. So what I'm going to do here is show you is that these things right here actually form a complex. So these guys are combined to give us a U4, U6. Now there's actually some base pairing occurring between these different snRNAs. And U5, where there's no base pairing occurring between U4 and U5 or U6 and U5. That's why we're using this little period right here in the name. So this snRNP, the U4, U6, U5 snRNP, there are 15 proteins, unique proteins, that are specific to that snRNP. And then there are 14 different ring proteins. 7 that are specific to U4 and U5, and 7 that are unique to U6 to give us the 14. So these are massive. They can be massive complexes with lots of different subunits. So, okay, now we have an idea that these can be somewhat complicated and have lots of different protein subunits. Why do we care? What's the point? So essentially, these all are going to be major components of the spliceosome. major components of the spliceosome. Now that spliceosome, what is that? That is the, it's a RNA and protein complex that is in charge of splicing the exons. So in charge of exon splicing. And it goes through a number of different changes so that there are, or I should say stages or complexes. So we'll have the E complex, the A complex, the B star, C, and then the post-spliceosomal complex. So a number of different complexes where the types of snRNPs and the non-USNRNP spliceosomal proteins together, the number and the combination of these are changing through each complex. But as the spliceosome goes through these different complexes, we're going to have those exons spliced together. I'm going to talk more about that in a moment. Okay, so that's the introduction. I've got some new we've gone over some new terminology, and now let's go over how this process works. So exon splicing. Now, a lot of people, even people who have been working in this field for a long time, will call it intron splicing, but that doesn't quite make sense, right? Because to splice something is to take two things that aren't connected and to connect them together. And I think when people use the word intron splicing, what they really mean is introns were spliced out, or introns were removed during exon splicing. So this isn't quite right, but you'll see that used in papers sometimes. It's really exon splicing. Introns are removed during exon splicing, but it's very common to call the whole process intron splicing too. But exon splicing, joining the exons together, is the more accurate term. Okay, so here is RNA pol 2. I'm going to stick with green for a little bit here. Here's my DNA, and it's unwound behind the polymerase, inside the polymerase there, but you can't see that it's unwound. I won't diagram the C-terminal domain with the heptapeptide repeats. I won't diagram that. I'll just diagram the nascent RNA. And what I want to go over now are the cis-acting sequences that are important for exon splicing. So cis-acting sequences, what that means is, these sequences are part of the nascent RNA. They're actually part of it. They're not found anywhere else. They're part of the RNA. So we have the 5' splice site here and the 3' splice site. Those little hash marks there, those are marking or marking the borders of the intron. Let's say this is the intron between those vertical lines. So the 5' splice site, most introns are going to have a G and a U at the beginning of the intron, and that's the 5' splice site, or the consensus sequence of the 5' splice site. And introns usually end with an AG, so that's the 3' consensus sequence, or the 3' splice site consensus sequence. So there will also be a branch point, which is usually rather close to the 3' end. So that is going to be called either the branch point or the branch site. And then in between the branch point in the 3' splice site, there's usually a polypyrimidine tract. And those are the major cis-acting sequences that are involved in exon splicing. So around each of these important regions, there are other sequences. So there are going to be some consensus sequences that are commonly found associated with these parts of the intron. Also within the intron, there may be some consensus sequences too. So other sequences that can help guide this process. But we're only going to focus on these specifically: 5' splice site, 3' splice site, and the branch point. We won't actually talk much more about the polypyrimidine tract. Okay, so those are the cis-acting sequences. They are part of the nascent RNA, and they're important for exon splicing. So now we have these transacting factors. And I don't know where these terms came from, cis and trans, and I think I did at one point. But usually they're very common in genetics and molecular biology. And cis we usually use for DNA sequences that are being acted on by some sort of protein, and trans is used for the proteins that are acting on the sequences. So the transacting factors, they're acting on something. In this case, they're acting on the nascent RNA. And the transacting factors, we've already went over these guys. These are the snRNPs plus the non-snRNP spliceosomal proteins. And so we already know there's many proteins associated directly with the snRNPs, but there are also some spliceosomal proteins that are not technically part of the snRNP. And all of these are going to be important. They're all the transacting factors that are needed to remove that intron and splice those exons together. Okay, so how does this process work? So let me find I'm going to use blue. Let's go with blue. So I'm going to start with a simple diagram of the mechanism. And this little rectangle right here, that's going to be an exon, the end of an exon, and this is the end of an exon over here. And so this right here, that's going to be my intron. So I wonder if I can change the opacity here. And let's see, I'm going to put an N right here. So we don't really care what that nucleotide is. But here, that's where our GU is going to be, right there, for the 5' consensus. AG back there. And let's say our branch point is going to be right here. I'm going to put circles around these here. So I'm not going to indicate the sequence of any of the other nucleotides there. So there are going to be some bonds that are important that we're going to need to look at here. So let me re-diagram this, change that opacity back to 100%. So I'm going to show one phosphodiester bond here. Just the phosphodiester bond between the N and the G right there. So every nucleotide here has a phosphodiester bond between it. I'm only showing one. So I'm going to show that G there. And why not? I'll show the U. And I want to show the phosphodiester bond at this side as well, between this G and the first nucleotide of the exon over here, the next exon. So I have N here, and branch point, I'm going to put the branch point here. Okay. So the diagram's getting a little messy, but I think you can see what's going on if you were paying attention. I'm just showing two phosphodiester bonds, one here between the exon and the first nucleotide of the 5' splice site, and one phosphodiester bond here between the last nucleotide of the intron and the first nucleotide of the flanking of the next exon. So the next thing to happen, so the first step in splicing these exons together involves the 2' hydroxyl on the branch point nucleotide, which is usually an adenine. So, okay, remember, these are RNA molecules, right? So what does an RNA look like? An RNA nucleotide look like? So there's the nitrogenous base up there. There's the 5-carbon sugar right here. There's the 3' carbon and the 2' carbon. The 3' carbon is connected to what? A phosphodiester bond connected to the next nucleotide, right? That 2' carbon has a hydroxyl group. So that oxygen attacks this phosphate group right there. So that's the first step. The oxygen on the branch of the 2' oxygen on the branch point adenine there attacks the phosphate group near the 5' splice site. So what that does, it gives us something like this. So it gives us a OH group there, and gives us, what's the best way to diagram this? G, I got the G here. I'm going to show that phosphate still. So we got that new bond forming here. So here's that O of that up there, up from the 2' hydroxyl group on the branch point nucleotide. So now that's connected to the A. There's the U that was next to the G, so this U up here is now here. And see how this U is connected down here to this A? Well, still connected to that A, right? And nothing's happened at the 3' end, so I could just copy the 3' end. Okay, so we have this loop formed now, right? So here's the branch point at adenine. The 2' oxygen, the 2' carbon, is now connected through a phosphodiester linkage to this G, which was the G of the 5' splice site. There's the U of the 5' splice site. And here's the rest of that intron right here. So we got this loop that formed. It's almost like a lasso, right? So that's called the lariat, or the intron lariat. So the first step forms an intron lariat. And these aren't connected here. No connection there. But that's okay, they're being held, everything's being held together by the spliceosome. And step 2 is going to happen like right away too. So step 2, I should probably be using more than one color to make this clearer. So step 2 is going to be this oxygen here is going to attack this phosphate group down here. So what that's going to give us, essentially, it's going to ligate the exons together. And you know what? Why don't I put the phosphodiester bond between these? So that's just the phosphodiester bond between the two exons, showing us that we've ligated the two exons together. When this O here attacks that phosphate group there. Okay, so we got 4 O's here, right? 1, 2, 3, 4. But there's 1, 2, 3, 4, 5 here. So where does that other O end up? Well, it just ends up right here on that G of the 3' splice site of the intron. So one product is going to be this on the left, the ligated exons. The other one is going to be the intron lariat here. So here's that branch point A, which is linked through the phosphodiester linkage to the G of the 5' splice site. There's the U of the 5' consensus sequence. There's this right here is all the intron sequence that was to the left of the branch point, and over here this is all of the intron sequences that was to the right of the branch point. And we're going to end with an OH group right there on the 3' carbon of this last nucleotide, whatever it was. So what was that last nucleotide? We know that, right? It was a G, right? Because the consensus sequence says their 3' consensus sequence is AG. So it's most likely an AG. Okay, so there we have the two different products of this mechanism here. We've removed the intron, and we've ligated those exons together. Okay, I hope that wasn't too confusing. Just one more thing to go over. And I've already diagrammed some of these for us. Let's see. Okay, so as I talked about in the beginning of this video, is that the spliceosome goes through a number of different complexes while the exons are being spliced together and the intron is being removed. So I've got some diagrams, and these diagrams are based on a diagram in the set of lecture notes that is available on ReggieNet. So the first complex that the spliceosome forms is complex E. So here we have complex E, and complex E forms when the U1 snRNP binds to the 5' splice site. And see, the U2 snRNP right here, what this does, this actually binds to the branch point and to the 3' splice site. So the branch point for sure, probably also to the 3' splice site. So my diagram isn't the best for indicating the binding of the U2 snRNP to the branch point. Complex A is formed when U1 and U2 come together, so bringing the exons closer together. Complex B is formed when the other snRNPs enter. So we see interactions between all of the snRNPs. So when complex B transitions to complex B star, well, U1 and U4 leave the complex, and that gives rise to complex B star. And it's in this complex that the branch point attack happens on the 5' splice site. And because of the diagram, we can't see that this exon here has been disconnected from the intron. But after the branch point attack and separation of the 5' exon from the intron, that's complex C. So complex C, within complex C, we have exon ligation. So that means that that 5' exon, that hydroxyl group there on the end of the 5' exon, attacks the phosphate group at the 3' splice site. So we get something like this. We have the exons ligated together. We still have the snRNPs associated with the exons, and we have them bound to the intron lariat. And this is called the post-spliceosomal complex. And then after that, everything dissociates. We have the spliced exons. We have the lariat that is released. And so the branch point would be here. We have the A, then we have the G, then the U, then the A, and the G down there. And then the snRNPs are released and recycled. So complexes they can be, there's tons of details that we didn't go over in structural things. But I just want us to sort of have an idea of, how these different snRNPs sort of move through the different stages of the different complexes of the spliceosome. So one more thing I just want us to kind of have an idea about, what's going on during intron removal and how these, say, snRNAs are, using. Why does a spliceosome need RNA particles? Why doesn't it just use proteins? Well, maybe I can zoom in a little bit here. It's a little pixelated. But I just want to show you that there's some base pairing that goes on. So nucleic acids are, can base pair with other nucleic acids. So we can see this U1. I just have a little diagram here of U1 interacting with the 5' splice site of this intron. And so here's an exon over here, and here's an exon over here. And so we can see why there's an advantage of having a snRNA, or having snRNAs, having RNA molecules participate in this process, because base pairing can happen. And you can use base pairing to sort of guide, where the splice sites are. So we have some base pairing going on here. All these little hairpin structures right here. So there are some, this is all important secondary structure of the small nuclear RNAs. And you can probably bet that the proteins that associate with the snRNAs are interacting with these various hairpin loops. And then here we have the U2, a model of the U2 snRNA interacting with, this is going to be the branch point right here, and the interacting with the 3' splice site, 3' exon, I should say, also. So we can probably guess that mutations at certain points of the snRNAs would interfere with the ability of the spliceosome to remove introns if those mutations interfered with the ability of the snRNAs to interact with consensus sequences within the intron by base pairing. So I think that's it for splicing. So if you have, hope you understood all of that.